So I'm Samuel Duodu, as was introduced, and I'm from the West Africa Center for Cell Biology and Infectious Pathogens at the University of Ghana in Accra, Ghana. So this afternoon, thank you for the opportunity, and I want to share what we have been doing for the past 5 years on trying to improve on the diagnosis of febrile illnesses in Ghana. So the topic for today is Multiplex panel for the accurate diagnosis of non-malarial febrile illnesses. Next slide, please. So as we all know, febrile illness is one of the common reasons why people seek medical care in Africa and also other parts of the world. And the accurate diagnosis of non-malarial febrile illness remains a big challenge in many malaria endemic countries like Ghana, in the sense that the causative agents or what is actually causing febrile illness is not often identified due to the non-specific clinical presentation and also the diagnostic challenges or limitations. We have a significant portion of febrile illness are usually assumed to be malaria and treated as such. And these actually contribute to most of the drug resistance that we see and also poor treatment outcomes. Next slide. Now, so this presentation actually is going to focus on the use of multiplex diagnostic tools for identifying pathogens associated with non-malarial febrile illness in children living in Ghana. Next slide. So this is what I said. We have been working on febrile illness for the past 5 years. We started in 2016, where we're trying to understand the etiology of febrile illness, because we know malaria is reducing. The bed of malaria is reducing, but we still have a lot of cases coming to the hospital. So we're trying to understand what actually caused, what is behind these increase or the maintenance of febrile illness, even though malaria is transmission or the bed of malaria is reducing. So we did a cross-sectional study, which is hospital-based study from two places in Ghana. Kintampo is in the middle belt of Ghana. It's some kind of a semi-urban town with a population of about 40,000, and also in Accra, from a place in Accra, which is a peri-urban area. So these two areas have different ecological settings, and the people living there have different lifestyle. So it was a good base to collect samples from. And also the transmission, the malaria transmission intensity is quite different. Kintampo is a high transmission zone for malaria, with I think infected dose. I mean, the biting, they have about 300 bites per person per year. And Accra is a low transmission for malaria, which has an effective biting rate for mosquito around 50 or less than 50. So we collected samples from children between 1 and 15 years who comes and present with acute fever to the hospital. And acute fever, we mean that those that come with temperature less or equal to 37 or 38, and the condition occurring less than 7 days. So we collected a lot of samples. We have more than 1,500 clinical samples from these two areas. Next slide, please. So normally, when a child comes with fever to hospital, it comes with a non-specific clinical presentation, either vomiting, diarrhea, abdominal pain, which is a very common syndrome or symptoms of fever. So it's very difficult to know what is actually happening to the child. So the first thing they normally do is to do a malaria test, because we are from a malaria endemic area. They do malaria RDT, also microscopy, to rule out malaria. And if, depending on the signs and symptoms, the clinician can also, diagnose other diseases like, next slide, please. Like requests for other tests like bacteremia, or probably look for urinary tract infections or gastroenteritis. And these are also, they collect blood, stool samples, urine samples, and they culture these samples to look for pathogens. And also, next slide, please. Also, the use of non-malarial RDTs are also common. I know we have RDTs for hepatitis and also HIV and others. Next slide. So from our study, I mean, these figure just shows, I mean, what was clinically diagnosed and what was actually confirmed in the laboratory to look for the pathogens. And if you look at the malaria, I mean, it's not quite impressive, but at least most of the clinical diagnosis were actually confirmed. So they were able to detect parasites in the blood of these patients, of these children. But if you compare to the rest, I mean, the urinary tract infection, bacteremia, the proportion of what was confirmed was very small, very small, very small. Next slide, please. So if you look at the different pathogens that were found in the urinary tract infections, bacteremia, gastroenteritis, it's a whole broad of pathogens. We have E. coli, strep, and all those coagulase negative staphylococcus, and also for the gastroenteritis, we have Giardia, Hemings, and all those were found. So it's quite a broad pathogen. So we can say that some of the kind of fever probably come with so many infections coming from different pathogens. So it's a polymicrobial infection that we are seeing here. Next slide, please. And we also did serological tests to see whether we can detect these pathogens. And surprisingly, we found a lot also in there. We found Salmonella typhi, Brucella, and others. Okay, hepatitis C, HIV, all those. So which still confirm that fever illness is a polymicrobial infection indeed. So if looking at the test, next slide, please. So looking at all these tests that we do, I mean, if we want to do individual, it's going to be very cost ineffective, and that means it's going to involve a lot of resources and also labor. And we are living in a very constrained environment, where resource limited. Our diagnostic infrastructure is not as good. So if you want to do all these individual tests, it's quite going to be difficult. So our study was trying to do a multiplex screening of this pathogen using what we call a TaqMan Array. So this TaqMan Array is a PCR-based assay that we detect individual targets, viral and bacterial targets, and also protozoa. So the TaqMan Array is a rectum-based assay which detects 3 protozoans, malaria, and others. It can detect 7 bacterial agents, and also 16 viruses. And you can actually run 6 samples in a single run. So we tried to do. I must say that this was not a very comprehensive study, but I mean, we're trying to understand whether we can use these approach. So we only took 166 blood samples for the screening. So what I'm presenting here is a snapshot of what is supposed to be there. And from our analysis, next slide, please. We found a lot of pathogens. We found, of course, Plasmodium was the dominant pathogen that was found, about 37.9%, almost 40%. But we also surprisingly found dengue virus. We also found Salmonella, Rickettsia, Coxella, HIV. Dengue has never been reported. As I think we discussed before, it's highly underreported. So, but with the TaqMan Array, we were able to fish out dengue for the first time. And that was the first reports, a molecular evidence to show that dengue is actually is being transmitted or in circulation in Ghana. So we reported these to the Ghana Health Service, the ministry in charge of health issues in Ghana, and they took that very seriously. So Rickettsia and Coxella is also not a normal routine. We don't have a normal routine diagnosis for these pathogens, and using this approach, we're able to detect them. These are all zoonotic pathogens. What I want to say is that a large chunk of the samples we cannot detect, we cannot assign any pathogen to it. So 57%, there was no pathogen detection, and these could be because this is a targeted PCR, so we have specific parameters approach targeting these pathogens. So it's some kind of a restricted analysis. I mean, so it could be that there are other pathogens also in the sample that we haven't actually made room for. So because our specific our probes and also the parameter that we use for the TaqMan is very restricted to these 26 pathogens that I indicated in the previous slides. Next slide, please. So we see the potential for the TaqMan Array that is doing something good. But one thing about TaqMan Array is that it's a bit expensive, and you need a well-trained personnel to handle that. And the Rita machine is not something that we find in every diagnostic lab in our hospitals. So we're thinking of something very feasible, something that we can actually use without much problem. So there's another study that we do, which revolve around multiplex peptide array. So this is a serological assay that goes to detect also multiple pathogens. So we have a microarray peptide chip developed, and I might say that this was a project that was developed in collaboration with TIBA in Edinburgh. So also a customized microarray peptide chip was designed with the 26 known febrile pathogens that was used in the TaqMan Array, and these peptides were spotted on the array chip. And we designed both IgM and IgG-based serological screening, so I can know whether what we detect is actively being transmitted or is just due to exposure. So with this peptide array chip, what we're doing is that we are profiling the circulating antibodies in the blood, and in that case, in a blood spot. So that means we need very small volume, which is an advantage in this sense, because for the TaqMan, you need at least 2.5 ml of blood to have a total nucleic extraction. But with these, get a blood spot. So the assay is still being developed. We have made our chips ready with the 26 pathogens, and we are still optimizing the system to detect these organisms in our clinical samples. Next slide, please. So the finding so far from this work is that there's a detection of multiple target bacterial and viral pathogens in a single biological specimen. So it's a polymicrobial infection that we see with these febrile cases, and our assay is able to detect them. And there's also growing evidence for the widespread occurrence of zoonotic and arboviruses in febrile patients, which is something good to know. There's a critical need for differential diagnosis for effective management of childhood, because it's not everything that comes to the hospital is malaria. As we can see here, we have other different pathogens that are involved in the febrile case that we see. So there's a critical need for the differential diagnosis if we want to effectively manage and treat our children. Now, digital molecular diagnostics have a very important role here, and if these can be coupled to that and deployed in remote settings in Ghana, it will be very useful. So Aubrey, we are still, we are working together on this consortium, and I hope we can provide clinical samples for testing when the listening is ready. Next slide, please. So at this point, I just want to acknowledge, I think Aubrey have done that before. We are part of the digital diagnostic for Africa Network, and I want to thank Professor Gordon Awandari, who actually leads this febrile study project in Ghana, and also the PhD and postdocs who have been working on this project for the past 5 years. Thank you very much for listening.